for our next session, which is on science at the beginning of HIV/AIDS. And our two presenters are Dr. Victoria Harden and Dr. Robert Ball. Dr. Harden is the retired director of the Office of NIH History and the Stettin Museum, an office she created during the 1986-87 observance of the NIH centennial. She has published on the history of the NIH and medicine in the United States, and in 1989 and 1993, she organized conferences on the history of AIDS, the proceedings of which became publications, AIDS and the Historian, and AIDS and the Public Debate, Historical and Contemporary Perspectives, sorry. She conducted over 40 oral histories related to AIDS, which were made publicly available in 2001. She received her PhD in American history at Emory University. Dr. Robert Ball has been introduced before this morning, but it gives me an opportunity to add to his introduction to point out that he is currently serving as the Vice President and President-elect of the Waring Library Society, a Friends of the Library organization supporting our activities. So every time you're on, Dr. Ball, I'm just gonna add something else, something like that. So now I'll just turn it over to Dr. Harden and Dr. Ball. I'm not sure if you determine an order, if Dr. Harden's going first. Or
ladies first.
Okay, I think I'm unmuted.
Yep.
And let's see if I can share my screen. Is this from, okay. All right. Is this okay? Everybody
can see it. Yes, thank you.
If there's any way to move our little pictures on the right, there may be images that'll be covered up otherwise. I don't know if there is any way to do it, but
it looks like the whole slide is there. So
yeah, the whole slide is there. It's just that some images may end up underneath.
Okay. Okay.
Dr. Harden, there is a way that you can hide your view bar for the functions and also the view of the participants.
Okay, except that I don't know how to do that. So unless the folks who are the experts can do that, I'm just going to move right along. 10 years ago, in 2012, I had the distinct honor of presenting the Joseph I. Waring Lecture here at the Medical University of South Carolina. And that's when I first met Dr. Ball. He and I now, as well as some other presenters at the symposium, have gray hair. And we may not, I hope this is not true, but we may not be around in 2031 to mark the 50th observance of the early years of this pandemic. And for that reason, and also as a daughter of the South, I salute the Medical University of South Carolina for organizing this event. Dr. Ball and other speakers this morning outlined the problems with responding to HIV/AIDS when it was viewed still as a disease of marginalized groups, and no one really wanted to talk about it. In my talk, and for which I always use text because if I start ad-libbing, we'll be here all afternoon, I want to examine the response of the biomedical research community, primarily in the United States, during the first 15 years of the epidemic. In that short time, biomedical researchers identified the cause of the new disease, developed a diagnostic tool, and found a therapy that converted the disease from a certain killer to a manageable chronic disease. And I note in the image I chose for the title slide is an image of HIV. This is the way biomedical researchers thought about AIDS. This was a virus they were dealing with. There are many twists and turns in this story, so today I'm only going to try to skim across highlights. I want to set the background first. Within the world of biomedical research, the work of many decades had begun to come to fruition in the 1970s. For immunologists, the ability to characterize the cells of the immune system and describe their actions was in its infancy, and was one of the most exciting new intellectual fields in medicine. Identifying the basic work of B cells and T cells in the body, especially the key role of helper T cells, was of key importance when AIDS appeared. Now, if we have immunologists in the audience, you may recognize just how simplistic this diagram of the immune system appears now, even though when it was published in 1983, it was aimed only at knowledgeable physicians. It was considered too complicated for lay people. Similarly, this chart shows that only in 1970 were restriction enzymes introduced, allowing virologists to cut DNA and recombine it. Genetic sequencing of viruses commenced in 1970, and in 1973, the first animal gene was cloned. Cloning and sequencing would be critical to working with the AIDS virus, but PCR was not there to help until 1983. Add to this the advent of computing in laboratory research, beginning in 1962 with the Link computer, the first laboratory personal computer. Molecular immunologists in particular needed computers to analyze data that they were obtaining as they began to sort immune system cells under the microscope. And one textbook in 1980 opined that for such an analysis, an entire computer that had 64 kilobytes of core memory and a disk that would store at least 10 megabytes would be required. One size, thinking about how much more power our phones have today. I hope that this background may remind you where biomedical knowledge was about viruses and the immune system, and the technology available to address the problem stood when HIV/AIDS appeared. Week by week in seminars and journal articles, researchers were learning brand new facts about these things at the same time they were tasked with responding to this deadly disease. After the CDC had ruled out known pathogens as the cause of AIDS, and epidemiological data pointed to an infectious microbe, research to identify the causative pathogen began. In the United States, most of the early research on AIDS took place in the laboratories and clinics of the National Institutes of Health because these laboratories had the authority to change the focus of their research overnight. Talented academic researchers across the United States, whose research was funded by NIH grants, did not have this authority. They could not abandon the work for which they were funded and turn to a new problem. Now, after Rock Hudson died, Congress made, in 1985, Congress made more money available to fund grants for AIDS research, and the 2-year grants process could be implemented, which means that money got out across the United States sometime around 1988. But before that, a lot of people's hands were tied. Groups in New York and at the CDC had noted that some aspects of AIDS looked similar to feline leukemia virus, a disease of cats caused by a known retrovirus. Now, before 1979, retroviruses were known to cause diseases in animals, but pathogenic human retroviruses were believed to be non-existent. Now, curiously, we can thank the National Cancer Act of 1971, also known as President Richard Nixon's War on Cancer, for funding that supported, under one part of the act, research into viruses as potential causes of cancer. At the National Cancer Institute in Bethesda, Robert Gallo's Laboratory of Tumor Cell Biology conducted research looking for a pathogenic human retrovirus. In 1979, Gallo's lab, along with a Japanese laboratory, demonstrated the existence of a single human retrovirus that caused adult T-cell lymphoma. And in 1980, a second retrovirus in that family was identified. Now, had these pathogenic retroviruses not been demonstrated to exist, we would most likely still be looking for the cause of AIDS. In Gallo's lab and in other NCI labs, all the retrovirologists that subsequently were involved in AIDS research were trained. When the search for the cause of AIDS turned to retroviruses, two groups were in the forefront. There was Gallo's lab at NCI and the laboratory of Luc Montagnier at the Pasteur Institute. Montagnier himself was not a retrovirologist, but he had hired Francois Barré-Sinoussi and Jean-Claude Schermann, who were. In 1983, the group at the Pasteur published a report of finding a retrovirus associated with an AIDS patient. They named this virus LAV, or lymphadenopathy-associated virus. They did not claim or attempt to demonstrate causation. In 1984, the Gallo lab published 4 papers demonstrating that AIDS was caused by a retrovirus, which they called HTLV3, because they believed it was the third in the family of retroviruses that the lab had identified previously. Now, both groups utilized laboratory assays for the virus that they then converted to prepare diagnostic tests, and they submitted them to the US Patent Office. The French application was submitted first, but it was based on a portion of protein in the virus detectable only in the earliest stages of infection, and they did not present data showing that they had tried the assay on a wide population. The Gallo patent application was based on a protein detectable over the entire course of the disease and included extensive data showing testing of the assay. The Patent Office therefore awarded the patent to the Americans, but the French cried foul because their application had been submitted first. Now, of course, the real issue here was the money to be made from an AIDS diagnostic test. And this dispute was settled in 1987 at the White House, where Presidents Ronald Reagan and Jacques Chirac agreed on a formula to split the proceeds of these tests. This was the first scientific dispute settled by heads of state. Today, the French test is used only in France. The American test is used in the rest of the world. Another international brouhaha related to the name of the virus. As I said, the French called it LAV and the Americans called it HTLV3. In papers published between 1984 and 1986, References to the virus reflected which side of this disagreement the authors were on. French supporters called the virus LAV/HTLV3, while American supporters called it HTLV3/LAV. The two laboratories sequenced the viral genome and demonstrated that the viruses were identical. So the name issue was submitted to the International Committee on Taxonomy of Viruses. Now, not wanting to take either side, this committee chose a neutral name, the human immunodeficiency virus, or HIV, which is the name we have known it by ever since. A final naming shift came in 1995 at the first White House Conference on AIDS, when enough evidence had been accumulated that AIDS was actually the end stage of infection with HIV, that the formal name of the disease was changed from AIDS to HIV/AIDS. Now, this chart, which I pirated from one of Dr. Anthony Fauci's PowerPoints, will explain why this name change occurred. As data were gathered through tracking patients from an estimated time of infection through death, it became clear that the AIDS virus killed helper T cells, the master cells of the immune system. This chart shows in blue the decline over many years, and note the broken lines here where we jump from 1 year, from months to years. And shows the decline of the T cells in a person's body. And in red, we get the rise in the amount of HIV in a person's blood. After the initial infection, which was experienced as any viral infection, a headache, fever, muscle aches, there might be no symptoms at all for nearly a decade. The body fights back, but very slowly does it loses the struggle as the volume of T cells slowly decline and the number of virus particles in the blood slowly increase. The most devastating opportunistic infections and cancers, as you can see here, appear only at the point that the immune system is nearly non-existent. You see down here, it's nearly non-existent. Just beginning to get constitutional symptoms here. And by the time you get to the worst, you just don't have an immune system. This led the medical community to understand AIDS, not as it was thought of initially, but actually was the end stage of a long infection with HIV. Now, in 1981, when AIDS appeared, there was only one antiviral drug available, and it was for a rare herpes virus infection of the eye. Acyclovir for genital herpes earned FDA approval in 1982, but that was as far as antiviral drugs went when AIDS appeared. During the summer and fall of 1984, after the scientific community agreed that a particular retrovirus was the cause of AIDS, laboratory groups cloned and sequenced the viruses they had isolated, and by 1986, they had identified all 8 genes of HIV, even though they didn't understand fully what each one did. They were able to draw this chart of the virus's life cycle, which appeared in a 1988 special edition on AIDS of Scientific American. Now, if you can see the chart, first, HIV has to enter a human cell. Next, its reverse transcriptase enzyme converts viral RNA into DNA. And then the virus's integrase enzyme splices this viral DNA into the DNA of the host cell. That's here in the middle. When the DNA in that cell unwinds to permit protein synthesis to occur, long strains of new HIV proteins are made, and then they are cut at strategic points that allow them to assemble into individual new viruses. And then these new viruses butt out of the host cell to infect new cells. Now, each of these stages offered a potential point of intervention to halt viral reproduction if you could just find a drug that would intervene at that point. So the question was, what drugs might do the job? The first drug discovered that showed any sign of effectiveness was azithromycin, or AZT, which blocked the action of reverse transcriptase. Now, thanks to another provision of the 1971 Cancer Act, Burroughs Wellcome Pharmaceutical Company had facilities for screening drugs. AZD had been synthesized in 1964 as a candidate cancer drug, but it proved inactive, so Burroughs Wellcome put it on the shelf. But when it was screened again for potential anti-HIV properties, Burroughs Wellcome scientist Dr. Mary St. Clair found that AZT inhibited HIV in test tubes. And so the drug was sent to the National Cancer Institute, which quickly set up a clinical trial. You have to have also in your head just how desperate everybody was to find something at this moment. Dr. Samuel Broder, Robert Yarquin, and Hiroki Mitsuwa moved as fast as possible through animal studies and very preliminary clinical trials to demonstrate that AZT slowed disease progression. The FDA approved the drug for wide use in March 1987, which was the shortest drug development and approval time in US history. Very quickly, however, it became apparent that AZT only slowed the relentless progression of AIDS, that the disease developed resistance to the drug, and that the drug itself had toxic side effects. Burroughs Wellcome marketed AZT under the name Retrovir and set a very high price for it, believing that more drugs would soon be developed and wanting to recoup its investment. Year after year, however, only a few similar drugs appeared, all of which shared the toxicity of AZT. AIDS activists were livid that Burroughs Wellcome was profiting so greatly off the suffering of people with AIDS. They demanded that new drugs be developed, as if one could demand and throw money at a problem and a cure would appear. ACT UP, the most politically savvy activist group, staged dramatic protests. Among others were those at Burroughs Wellcome, at the New York Stock Exchange, and as you can see at this particular photo, at the NIH. Finally, in 1996, David Ho's laboratory in New York published data on drugs known as protease inhibitors, which interrupted the HIV life cycle at the point where new viruses were being assembled. On this more detailed diagram of HIV's life cycle, the protease inhibitors inhibit strings of HIV genetic material from being cut and packaged into new viruses. Physicians then combine the AZT type and the protease inhibitor type to make a cocktail that did indeed stop the virus from replicating, and voila, patients were able to get up out of bed and go back to work. At first, they had to take many, many pills a day, as this photo shows. But eventually, it was reduced to 1 pill a day, the therapy that continues to be used. This combination antiviral therapy literally transformed AIDS into a manageable chronic disease. AIDS patients who had made their wills now had to figure out how to re-enter society and pay their mortgages. It was a good problem to have. So to summarize, AIDS had been recognized as a new and deadly disease in 1981. And in only 15 years, An effective therapy transformed it into the chronic condition we call HIV/AIDS. No preventative vaccine has been developed, however, because HIV mutates so fast that classic type vaccines will not work against it. In medical history terms, this therapeutic achievement was breathtakingly fast. Think, for example, of plague, diphtheria, tetanus, smallpox, all diseases that killed millions of people over the centuries before either a vaccine or a therapy was identified. People with HIV must take drugs for the rest of their lives, and the drugs themselves have toxicities for the heart, kidneys, and liver. But as Paul Boberdein, one of the first physicians to treat AIDS patients, remarked about the introduction of what was initially called highly active antiretroviral therapy, or HAART, there is probably not anything equivalent in medicine apart from maybe the development of penicillin in terms of a night and day difference. Now, of course, for those whose friends and loved ones died during those 15 years, the therapy did not come soon enough. And I want to close with this image of the AIDS quilt, panels of which were organized by the NAMES Project in order that those who died of AIDS not be forgotten. The red ribbon also, I point out, that symbolizes AIDS was created in 1991 by a graphic arts company in New York called Visual AIDS as a simple symbol declaring solidarity among those living with HIV/AIDS. It inspired many later groups of health lobbyists, beginning with advocates for more breast cancer research, who adopted a pink ribbon symbol, which we may see more frequently today than the red one. Today, many other groups use similar ribbons of various colors. And with that, I'll stop. Thank you so much.
And I think Dr. Ball is up next. Robert?
Oh, let me stop.
Yep. Okay. Thank you, Dr. Harden. It's good to see you again. And thank you for taking us through a life trip through the history of HIV/AIDS. Let me share my screen here now, Brian. No infectious disease doctor can talk without slides. So that's a maxim here. And let's see if this will go full screen. Brian, is that full
Yes.
screen?
Yes, it is. Yep.
Great. In the beginning, there was some science, as Dr. Harden has shared with us. What if, and imagine, what if you were on the cutting edge of diagnosing a new infectious disease whose origin was totally unknown? This has occurred time immemorial through history. What tools would you use that were available at the time? This is one of the success stories of medicine, laboratory medicine in particular, of getting us there. So I'll move on. Next slide. Slow to advance. We saw this morning, South Carolina, first patient we had, and this was an interesting case because my infectious disease fellowship did not include much immunology. So let's look at the bottom paragraph. We know what he had. We know that his white cell and lymphocyte counts were extremely low. Whole blood was sent for lymphocyte testing. And what was found then, as I briefly mentioned earlier, a low OKT3 cell count. Interestingly, OKT3 actually refers to a an antibody on the surface of certain white cells that can be measured by flow cytometry in those days. It is now the equivalent of, or indirect equivalent of, the T4 or CD4 cell count. Likewise, there were very low suppressor OKT4 cells, now called T8. If that wasn't confusing enough, then there developed ratios of OKT3, OKT4, which are now we call CD4 ratios. Next slide here. Immunoscience at the beginning. Cell flow cytometry in the lower left, the earliest laboratory instruments used, basically measured through a tube flowing through past a light sensor, fluorescein tagged cells, particularly tagging monoclonal antibodies. And there at about 3 o'clock on the slide, you can see a graphic of labeled antibodies, and you can label different antibodies with different imaging chemicals. Next slide. This is fairly complicated, don't need to go into detail, but these cells actually measured a specific type of immunoglobulin antibody, if you will. And the CD3 antibody is part of a complex of antibodies found only on and in mature T cells and thymocytes. Interaction with the T cells causes T cell activation. So T4, the so-called helper cell, activates our T cells to respond to various invading pathogens. In the bottom paragraph, the cytotoxic T8 cell also plays a role, we'll see in a moment. And then these cells come and go rapidly. So I'll do next slide. We know about helper suppressor counts, CD4 white blood cells, helper cells, stimulate other immune cells. I like Dr. Fauci's description here, as Dr. Harden has referenced, but I like this one the best. The conductor of the immune system orchestra. Just imagine a symphony orchestra, as beautifully complex as our immune system is that God has given us, that there is a key cell in there that is the conductor. Now, the other parts of the orchestra can play their parts, but not in synchrony nearly as well. Now, the CD8 cells, suppressor, recognize and destroy some bacteria and viruses, but they also suppress CD4 cells. So hence the T4-T8 or CD4-8 ratio was an early mark of immune deficiency that worked up until the 90s when viral load testing came in. Next slide. As mentioned in a slide earlier, we've known about lymphocyte subpopulations for many years now, and Dr. Ahuja tomorrow may go more into detail. But at the bottom of his nice slide here, the goal is a viral load less than 20 copies per mil, which is basically indetectable. So undetectable now roughly equates to untransmissible, and that U equals U is important. Next slide. Discovery, as Dr. Harden has referenced, here's Dr. Montagnier and his colleague Francois Sissou, and another graphic on the lower right that is very useful. They received the Nobel Prize in 2008, but as Dr. Harden mentioned, where was Dr. Gallo in all this? He was key in early discovery of retroviruses. He actually was one of the first who discovered human retroviruses, human T lymphotropic virus type 1 and type 2. Type 1, the cause of specific leukemias, type 2, lymphomas, with some overlap between the two, and had continued in research, particularly at NCI, for some time. And likewise, as Dr. Arden mentioned, teamed up with Dr. Montagnier and others And they agreed that this retrovirus, HTLV3, they called it in some circles, LAV, and ARV. ARV is AIDS-associated retrovirus, all by the common names. And as mentioned, the presidents called a meeting to settle this. The first time politicians, national governments intervened really significantly in modern medicine, and they basically said, you're going to work together and settle this. And hence the HIV, the name HIV came out of that. But further research, the success story is that further research funding from national governments. Well, Dr. Gallo finally admitted that his virus probably came from France. So he was never awarded the Nobel Prize, but he continued academically active, contributing to many projects. Methods were cell cultures in those days, ELISAs for specific proteins, leading to genetic sequencing that we have nowadays. Okay, next slide. Here we go. More interesting developments. Here we see the nomenclature change, or nomenclature if you're British, and the International Committee developed HIV as the nomenclature. And about the same time, Dr. Kerry Mullis, who got his PhD in biochemistry, was working on DNA sequencing with primers, certain specific primers, able to say, we're going to select a certain segment of the genome and copy it in rapid sequence, and so that it doesn't get destroyed by the machine, the heat of the machine, and hence developed PCR testing in concert with other colleagues. And he got the Nobel Prize along with other colleagues, the co-Nobel Prize for PCR development. Dr. Mullis has an interesting history. I met him in Columbia. He grew up in Columbia, South Carolina, in the southeast section where Dr. Bryan and I lived for a while. Dr. Charles Bryan is still there, and some of our other colleagues are there. And he went to high school just down the street from where I live. I met him, and an odd fellow to say the least. He eventually ended up at UC Berkeley and was a surfer, used LSD, a very checkered history, shall we say. Anyway, now we move into molecular phylogenetics to determine the origins and dendrograms like we have seen recently with COVID. And the PCR sequencing can help determine some other HIV characteristics, such as resistance characteristics. However, we all know that without whole genome sequencing, WGS, it's probably going to be impossible regarding HIV and also hepatitis C virus to develop vaccines because of the mutant swarm, the quasi-species that develop from these viruses. Okay, and next slide. Sorry, it jumped. Okay, there we go. Lab tests in HIV treatment decisions. This came on not early, but in the process of finding out more about immunology and how HIV impacted. The CD4 count tells us, of course, about the immune system. Normal is around 500 or greater. In subsequent years, the Department of Health Human Services, in addition to the National AIDS Societies, infectious disease societies, et cetera, determined that you don't have to have an opportunistic infection on top of HIV, like a 1-2 punch. You just need to have a low CD4 count of 200 or less as an AIDS case definition. And that was a sea change in epidemiology in the definition of cases. In the middle here, viral load. number of copies, of course. And remember, this is a log scale, powers of 10. So one log change is a tenfold. And the goal now, undetectable viral load, untransmissible, we are all aware of that now. And these used to determine the CD4 counts and viral loads, used to determine the timing, when to start antiretroviral therapy. But the guidelines now give us the recommendation that in most folks testing positive, even with the rapid test on the sidewalk, healthcare professionals should try to start antiretroviral therapy right away and not wait on the CD4 and the viral loads. Otherwise, it's a missed or lost opportunity for many folks that we come across. Okay, and then moving on, I mentioned Department of Health and Human Services and collaboration with Infectious Disease Society, et cetera. This busy graphic here gives us one of many tables in their latest 460-page guidelines for adults and adolescent HIV, with HIV. And this table is laboratory testing. I'm going to focus in these few moments on laboratory testing. HIV serology, obviously, a rapid test should be confirmed by a venipuncture, blood going to a laboratory to confirm that. And in most cases, it confirms it. Certainly HIV-1 and most of the time HIV-2. HIV-2 has originated from sooty manga bays in Africa, and that's rare, usually in Cameroon. We know that SIV originated from African green monkeys, and HIV-1 originated from chimpanzees, Pan troglodytes, the genus and species Pan troglodyte. Okay, in the next row, CD4 cell counts. Yep, entry, and then repeating them every 3 to 6 months until you get viral loads that become undetectable. That is your goal. Then down here, resistance testing, entry into care, and after initiation of antiretroviral therapy, that is an important element of early care, which we will hear more about from our HIV infectious disease clinicians. And certain drugs like abacavir here, HLA testing and troponin testing. And the recommendations become more complex over the years and certainly beyond the scope of this short discussion. So one more slide, I think. Here we are. Here we have a few retrospective thoughts. At the start, I said, imagine, what if you were coming across a novel pandemic? You didn't know the agent, didn't know much about it other than it greatly suppressed the immune system. So here's some what ifs. What if HIV had occurred prior to discoveries of antibodies? Remember, antibody testing goes back more than a century. The 1906 Wasserman test for syphilis. Now we have the RPR and the VDRL tests. And antibody detection has become quick and easy with rapid tests almost on the spot. And now we even have rapid antigen testing for novel pathogens like COVID. Okay, second point, what if it occurred a few decades prior to us understanding the intricacies of the immune system? In infectious disease fellowship, when this gray hair went through, we had very little immunology. We had to learn it OJT. Third point, what about prior to laboratory discoveries of the mechanism of action of viruses? Medical science has known about viruses since the late 1800s with the tobacco mosaic virus, but the onset of many viruses such as HIV and others in the mid-1900s was indeed novel and had to be intensely investigated. Dr. Harden may correct me, but I think the earliest evidence of HIV in Homo sapiens goes back to in the NIH archives to 1921, but we can talk about that. And then what about HIV prior to the awareness of retroviruses? Retroviruses have actually been known for some time with in the early 1900s with HTLV-1 causing leukemias, particularly equine infectious anemia, horse disease. And then lastly, what if HIV had occurred a few decades prior to developing the PCR testing and its utility in detecting the viral swarms? I'm not sure if we will find a vaccine out of this, but we can certainly detect resistance mutants, and that will help in directing therapy, which has become amazingly simpler nowadays for patient compliance. And I think that's all I have at the moment. I will stop there and be happy to answer any questions.
Thank you, Dr. Ball and Dr. Harden. We do have a few minutes for questions and comments, so if you want to put them in the chat and I can keep track of them. I think it's interesting when you talk about the what if scenarios. It seems when you look at the broad evolution of the history of health sciences, there are examples of these patterns where there's great confidence in the medical world that we kind of have a handle on things in medicine, and then something happens to disrupt that confidence, and then there's a re-examination, but then there's this great advancement of knowledge that comes out of that event. At least that's my impression. I don't know if you either one of you would describe it that way, but I point out I look at the influenza pandemic in 1918. AIDS is certainly that way. COVID seems to have had kind of a pattern like that. Dr. Harden?
Yeah, I interviewed early on as I started my AIDS interviews, which I started, by the way, because nobody else at NIH seemed to care. I interviewed both Dr. Richard Krause, who had been the director of NIAID before. Then I interviewed Dr. Anthony Fauci, and I asked them both, what would have happened if AIDS had appeared in 1955? And their answers were very interesting. Dr. Krause said, I think we would have recognized just from the epidemiological data that it was a sexually transmissible disease, and we would have put public health laws into effect to try to control it. Dr. Fauci, who was a generation younger, his instant response was, we wouldn't have had a clue because we didn't know the details of immunology and virology that we knew. So you have the two different ways of approaching the problem.
Yeah, that's very interesting.
Amen.
Yeah, it's very interesting. You also pointed out the broader story of Gallo in France, if you wanted to explain that further.
Well, I spent a whole year trying to run down what exactly constitutes discovery in science. And there are a number of points to make. One of the points is that you have to publish. And the Gallo lab published, and the French lab never made any claim. But the central point with respect to the Nobel Prize, in my humble opinion, and I do not think the Nobel Committee is God by any stretch of the imagination, is Bob Gallo's personality. Okay, people either love Bob Gallo, they hate Bob Gallo, they do both. And I know Dr. Gallo, and I have interviewed people who cannot bear the man, but none of them question his brilliance. And the night I met Dr. Gallo, which was at an NIH party that my husband and I were invited to, I walked up to him and said, "Hi, I'm the NIH historian, and someday I want to interview you." He stopped right then and there in the hallway, and he started unloading on me all this heavy-duty science about what he had done to investigate AIDS. And I'm thinking, I don't have a tape recorder. Please shut up and wait until I can record you. It was the most intensive interaction with another human being I think I'd ever had in terms of intellectual interaction. But because of his personality, an awful lot of people wanted to see him as the villain. And this certainly came through with, well, let me step sideways too and say, I was very pleased with when Dr. McKay, in his book, he did not take the Randy Schultz book as received truth about the epidemic, but as a book with a point of view, because Randy Schultz's point of view was highly influenced, not only by Dr. Conant, but by Dr. Donald Francis, who had been at the CDC, and he was one of the people who absolutely hated Gallo and wanted to see him come down. Now, however it all plays out, this is why I say we folks with gray hair need to get our stories on the record, because we're going to be the primary documents for future historians who have less emotion invested in the early years and who, with luck, are smart enough to see things more clearly. And I'll shut up at this point. Dr. Ball, you may want to comment.
Wonderful recollections. And one time I met Dr. Gallo at a meeting, very briefly, thankfully. It was all about himself. I'll stop there. And you're certainly correct about his complex history. Most bright people have checkered histories. I see in the chat box an interesting question or set of questions from Dr. Enrig about Dr. McKay's work highlighting phylogenetics. And that's a complex topic itself. I think phylogenetic sequencing, molecular understanding, and understanding of certain genomes with the HIV quasi-species swarm will help in our understanding its pathogenicity and may, after we gray hairs are gone, lead to vaccines, which will, by definition, not be perfect. The virus will usually escape vaccines, just like COVID Omicron BA2 escapes now. Second question that Dr. Enrig asked about the role of science denialism. It is prominent nowadays more than ever before. We have had vaccine denialism for years. I just finished a ASM medical textbook novel chapter on pandemics. And interestingly, even Benjamin Franklin, who supported smallpox vaccination, variolation in those days, as well as using cowpox vaccine, failed to vaccinate his own son who died of smallpox. So vaccine denialism has been around a while, has increased and decreased, waxed and waned over the years. We saw it horribly 15 years ago to 12 years ago with the measles vaccination deniers and the measles outbreaks in this country and other countries. And now we're battling it even worse among science deniers. There's a nice t-shirt that you can get through northernsun.com that says, the universe is made of protons, electrons, and neutrons, and morons. Okay. And Dr. Inrig also asked, what impact would COVID science have on science responses to HIV? Again, complex question, and it has to do with immunology, understanding of genes, molecules that will help ultimately in both. Unfortunately, science deniers don't see that nowadays. There is a national undercurrent of anti-Tony Fauci resentment, even hatred among certain political segments that is just sad to see. I'll stop there.
Did you want to add anything, Dr. Harden?
I would basically just underscore everything Dr. Ball said. That I recently gave a talk on a history of vaccines, and actually anti-vaccination goes back to even before Jenner's vaccine to the smallpox variolation and all the way up. And for the first time in my entire career as a historian, a group I was asked to speak to asked me, would I mind removing references to anti-vaxxers because some people in their group like to make their own decisions? And I told them, no, I wouldn't do that. But my point is simply to underscore how incredibly strong some of this is. And it breaks my heart that people have no idea that Tony Fauci, I like to say, knows his epidemics. I mean, from AIDS to SARS to MERS to Zika to West Nile and on and on and on, Ebola. The man knows what he's talking about, and it breaks my heart that people are not listening to what can protect their health.
Right.
That's enough.
Right. And it's interesting to look at that long history. I know Dr. Ball brought up Benjamin Franklin. I know Franklin regretted that decision the rest of his life. He felt guilty that he basically had killed his own child by making that decision, and had regretted it. So thank you very much for your presentations and your comments to questions and other comments. Richard McKay had one final comment. So true, the oral historian's challenge, the best stories coming out when there's no audio recorder. So I think I'll leave it at that. We have our next session coming up at 2:15. So I will see everyone in about a minute and a half. Thank you again,